So it's a great pleasure to be hosting Thomas's talk today. I've known Thomas for a while now. I think we met when we were a postdoc at Berkeley.
Yeah.
So Thomas did an undergraduate degree at Indiana in maths and philosophy, then went on to do graduate degree in neuroscience in Minnesota, then moved to do a postdoc with Jack Gallant at UC Berkeley, and that's when we met. Then he moved to South Carolina, where he started his own lab, and recently, maybe a few years back, moved back to Minnesota, where he's faculty now, and he is working from there with his lab. Thomas has made significant contributions to the field of cognitive computational neuroscience. He's a pioneer in using very advanced techniques of voxel-wise encoding modeling, combining deep learning algorithms with functional magnetic resonance imaging to study vision, but also imagery. And the talk today will be brain-optimized network models of the human visual system. So as I mentioned, keep your questions for the end. And with all of this, thank you, Thomas, for coming to present today. And the mic is yours.
All right, thank you. And thanks for the invitation. It's great to be here. We'll talk about brain-optimized network models of the human visual system. I'll start with the human visual system and then get a little bit later into what brain-optimized network models are. So I want to start with a picture of a brain, of a human brain. Actually, I want to start by acknowledging my collaborators, Gislon St-Yves, who's here, and Kendra Kay, who's also at the University of Minnesota. And they did a lot of the work that I'll be discussing today. So I want to start with a picture of the human brain. This is a digital rendering of the cortical surface of a human brain. And I want to just reorient it and process it a little bit to give you a picture, to show you how we derive the picture of the brain that I'll be using several times throughout the talk. So I'm just going to rotate it a little bit and then inflate it, then cut it and flatten. And this is now a flat map of the two hemispheres of the cortex, and the particular chunks of cortical real estate that I'm going to be spending a lot of time exploring show up right there. And as you can see, there are four areas denoted by these different colors. They have what we believe to be our distinct boundaries, and they're usually referred to numerically. V1 for visual area 1, V2, V3, and V4. Now, when I give talks to audiences that are not entirely visual neuroscientists, sometimes a question comes up that I've struggled to answer, and it's the one that's going to motivate a lot of the work I'll be talking about today, which is, why is there more than one visual area? So I've just shown you 4 of the dozens of visual areas in the human brain, and this is true of primates in general. There are just many visual areas. And why don't we have just one? I mean, why can't we see and do vision with just one area? And I'm not going to be able to answer that question, but we'll kind of increment our way towards an answer. Part of it has to do with the fact that each one of these areas is its own distinct map of visual space. And I'll show you some of the data that lets us know this. So what I'm showing you here is every circle, this is fMRI data. Every circle is a representation of the visual field. It's a display field that subjects, human subjects, who participated in this experiment saw. And during this experiment, they're lying in a scanner, they're having their brain scanned, and they're seeing visual stimuli displayed on a screen. Right? And the kind of the area of the visual field that they're looking at is shown by this circle. Each one of these columns is a different human brain. So it's data this is from one subject, from another. Okay, so there are 6 subjects in this data set. And each row is data from a different brain area. So the 4 brain areas I just showed you are represented by the rows. What are these colorful dots? They are the locations of receptive fields for individual voxels in the human brain. So when you're recording brain activity with fMRI, the unit of measure, or sorry, the unit from which you measure brain activity is called a voxel. It's a three-dimensional pixel you can think of. And every one of them can be characterized by the position in the visual field where a stimulus will really drive the voxel to its maximum activity. Right? And if you plot all of those points that indicate the receptive field locations, for this subject in V1, you see a complete covering of the visual field. So some part of the brain is responding to stimuli everywhere that you might show it, right? And what I'm emphasizing here is not the difference between visual areas, but the fact that they all look the same, or roughly the same when you look at the data. This V1 has a complete map of visual space, so does V2, so does V3, so does V4, and this is one of the most consistent findings across subjects. All subjects with functioning visual systems show this kind of remapping of visual space over and over. Okay, so this is not an answer to a question why you have many visual areas. In fact, it just leads us to another one. Well, fine. Why are there so many maps of visual space in the primary visual system? Why keep repeating this representation of visual space over and over? The answer to that question has something to do with the fact that every one of these areas represents different aspects or features of the visual stimulus. So if we think of the receptive field of each voxel as a little window on a stimulus, and we draw them we draw these receptive fields being stimulated by this painting here. We can kind of summarize, or I'm going to kind of summarize some of what we know, a little bit about what we know about the representations in each of these areas. So we know that activity in V1 can be characterized to some degree of accuracy as a kind of simple filtering operation that's sensitive to edges. There's some data that activity in V2 is sensitive to which object and where the object that owns the edge is located in the visual field. There's some evidence that in V4, the geometry of these edges is a really important driver of activity. There's kind of confusion about what V3 is supposed to do. But as you can see from this cartoon, which is just a very superficial skimming of the literature on these different areas, each one of them is doing something different. Okay? They're sensitive to different aspects of the visual stimulus. Okay, we still don't have an answer, but we've refined our question at least. And that is, so fine, why are there diverse representations in the primary visual system? Why do you need all these different representations? And by the way, I just want to remind you, there are dozens of other visual areas, and there's a story to tell about what they represent as well. I mean, they're different from all the areas I just showed you. So one of the really, I think, influential and long-standing ideas that provides a potential answer to this question is the idea of a processing hierarchy. And it's a really central idea in visual neuroscience and in artificial intelligence. And I'm showing one of many pictures that I could have taken from one of many papers that shows this basic schematic, this outline of a computational model of the visual system. In this case, we have an outline of the macaque brain, which is a really commonly used model system for studying vision. And this is showing you where V1, V2, and V4 live on the macaque brain. It's also showing this vast swath of visually sensitive cortex on the ventral and lateral side of the temporal lobe. All of these areas are responsive to visual stimuli and process it in different ways. And the kind of default model, it's not especially controversial, of the way that these areas interact and the way that the representations in these areas diversify is this hierarchical processing one, or what I'll refer to as a processing hierarchy. So now the visual maps that I discussed are represented by these grids. The receptive fields are little circles in these grids. And what is being shown is the idea that the activity in each one of these areas is fed to it by the area below it on the hierarchy. And it is processed by a simple, relatively simple canonical function, which they're calling T in this case, and then sent to the area above. Right? And this is an idea that goes back decades to the work by Kubel and Wiesel primary visual cortex, and it's kind of been passed back and forth between AI and neuroscience for years. And in fact, this is the logic of many deep neural networks. It's the serial processing chain, the processing hierarchy. I want to, because this idea is so important, I want to take some time to think about what you're kind of committing yourself to if you accept it, or if you take it as a starting point or a summary of our knowledge of the visual system. So I'm going to draw a hierarchy. Each visual area is now a node. So this isn't a receptive field. I'm using circle to represent a processing stage in the brain. And I'm labeling them V1 through V4. And in our understanding, the stimulus would be at the bottom of this hierarchy. It's fed to the retina, and the cortical areas arrange themselves in this order, with V1 on the bottom of the hierarchy and V4 at the top. And the processing hierarchy idea is really an idea that the visual system can be modeled as a series of function compositions. So what do I mean by that? Let's say that we label the stimulus X, right? And according to the processing hierarchy idea, V1 activity can be read out by applying a function to X, f of X. And it has its own sort of local parameters, which we'll call theta 1. Now, V2, the activity in V2 can be read out by taking the outputs of V1 and applying that function again. Right? Again, there's maybe there's wiggle room here because it has its own set of modifiable parameters theta. And you see where this is going. V1 is f of X, V2 is f of X, V3 is f of f, and so on. All right. So this is kind of a basic conception of the visual of the visual system, at least the primate visual system. And as I said, it kind of, if you accept it, it makes certain implications that you may or may not know that you're making. So one of them is that it gives an explanation for how representations diversify, and it's they diversify by just repeatedly applying this function. It also generally assumes that the point of applying this function over and over and over, in other words, the point of having multiple visual areas, is to build incrementally towards the solution of some computational goal that solves a task. Okay, so you can imagine a hierarchy extending deeper into the brain, which takes you to a brain area whose representations make the solution of a computational goal effortless or easy to read out by other brain systems. Okay. The other commitment I think that comes with this idea is the assumption that the diversity of representations does not imply a diversity of functions. In this processing hierarchy idea, each brain area is like the rung on a ladder. Basically, its job is to get you higher up on the ladder, where you'll encounter another rung, and then you're going to step on that, and you're going to get. But the point of the whole ladder is not to climb rungs; it's to get you somewhere, right? It's to get you higher. Finally, this, one of the kind of hidden assumptions of the processing hierarchy idea is that, this implies a constraint on local computation. And let me just show you what I mean by that. The take home is that hierarchies are needed only if local computation is relatively simple. So let's just assume a toy system whose goal is to compute x to the fourth. Right? And let's say in order to compute it, you have access to some processing stages, but the only thing they can do is compute x squared. If that's the case, you're going to need a hierarchy with 2 stages to compute x to the fourth. Right? However, if in each stage you can compute x to the n, you only need one. You only need one stage. All right. So this implies, if you think about this, the processing hierarchy idea implies that if we had more talented neurons, we wouldn't need so many brain areas, because we could represent whatever is supposed to be represented at the end of this chain with one synapse. Okay. Now, there's evidence that neurons are actually really talented. There's a recent paper that I think argues this pretty convincingly. So this is a first kind of point of criticism or skepticism I'm going to introduce to when thinking about the processing hierarchy idea. That said, there is a lot of empirical evidence that it can be construed as supporting hierarchy. And I'll walk through some of it. So a really classic piece of evidence is a notion of hierarchy that you can define by looking at pairs of brain areas, say V1 and V2, and identifying the reciprocal connections between them, the layer in which the reciprocal connections between them terminate. And by layer, I'm talking about cortical end. Right? So as many of you probably know, the cortex has 6 layers. And Fellman and Van Essen pointed out that if you look at pairs of areas and you say, okay, area A is the one that sends axons to layer 4 of area B, and area B sends axons to any layer but 4, you can use this as a rule to say that B is higher than A. And if you have tons of data which they access on the axonal connections between brain areas, you can construct this complicated looking diagram that really has a very simple summary. Okay. The hierarchy that they constructed by applying this simple rule is on the y-axis. Hierarchical ordering is on the y-axis here. And by applying this rule, you see that V1 shows up at the bottom, then you have V2, V3, V3A, V4, and so on. That's one really well-established notion of hierarchy. There are many others. So there's an idea of path length, which is just count the number of synapses between the retina and whatever brain area you happen to be in. Okay. And I mean, it is the case that many of these areas are several synapses farther away from the retina than others. So there's some diversity there. Brain areas are causally dependent on one another, which implies a hierarchy. If you cool V1, activity in V2 and V4 more or less vanishes. So there's clearly some kind of causal dependence. The time at which neurons activate after you present a stimulus, it's called the onset latency is widely distributed, and the times are shorter for V1 and longer for V4. That's another form of hierarchy. Also, as I showed earlier, or actually I haven't shown this yet, but I will, receptive field sizes get larger. You go from V1 to V2 to V3. I'll show that in a moment. And finally, as I implied in an earlier slide, representations somehow become more complicated. You go from representations of simple edges to things that are closer to objects, things that we can name. Now, each one of these is consistent or supports some notion of hierarchy. None of them really perfectly support the processing hierarchy idea that I've defined. I mean, there's some of them are consistent with it. In particular, the laminar idea, or the Fellman and Van Essen idea of hierarchy, is totally unclear to me at least how that relates to the simple processing hierarchy that I've defined, this series of function compositions. But recently, there has been some evidence that I think provides what at first pass at least is some pretty strong evidence for processing hierarchy in V1 through V4 and beyond. And that is the alignment of visual areas to task optimized deep neural networks. And a number of people have shown this. Okay. So what do I mean by alignment? And what do I mean by task optimized deep neural network? So here's a picture of a deep neural network. In general, when I'm talking about artificial hierarchies, I'm going to use trapezoids. So when I'm talking about deep neural network, I'll use trapezoids for the layers. And what I'm illustrating here is a system that's been trained to map stimuli, pixels, to labels, to object labels. All right. So this system is task optimized in the sense that its task was to recognize objects and images. And if you give it tons of labeled images, thousands of labeled images, maybe millions, it can learn to do this task very well. And this is one of the results that kind of launched the deep learning revolution right around 2012. Now, if you take this deep neural network, it's been trained, it hasn't seen any brain data at all. All it's seen is images that have been labeled. And you train it, and then you use it as the backbone for predicting brain data. In other words, for reading out brain data. What you find is that brain activity in V1 through V4 and beyond, all the way up and down to T, can be very accurately read out from the layers of this neural network. And by read out, I just mean you can apply a simple linear regression or a simple readout rule to the activity of the artificial units in these layers in order to get an accurate prediction of how these brain areas will respond to basically any stimulus you want to show. And you know that it's accurate because you have presumably some brain data that you've harvested from a human or an animal model, right? And you can show those pictures, record brain activity, and then make these predictions if you have a trained neural network model. The other really interesting finding is that the ordering of brain areas that we infer from basic neuroscience results matches the depth of layers in these neural networks. Okay, so what I'm showing here in this cartoon is that the layer that most accurately predicts V1 is below the layer that predicts V2, which is below the layer that predicts V3, and so on. Right. So there are a lot of papers that have data along these lines. I'll just show you some data from our lab, which is unpublished, but it makes the case well. So here I'm going to show you why I'm stating that these deep neural networks permit really accurate readout of brain activity. I'm going to show you a map of a human brain, and I'm coloring this map according to how accurately brain activity at each one of these points on the cortical sheet can be read out from a task-optimized deep neural network. And just to show that this is a good model in some sense, I'll compare it to kind of the last generation model, which was a model based on relatively simple filtering operations. And you can see there's just a lot of less gold and white on this map than there is there. By the way, this color bar is in units of Pearson correlation. White means really accurate, and 0 means no predicted value at all. And you can see that the deep neural network model gives you accurate predictions kind of very deep into the brain. It kind of highlights every corner of the cortex that is visually sensitive. If you take each one of these vertices on the cortical sheet, and on the y-axis, if you superimpose these models, you can kind of see the advantage of the deep neural network relative to the Gabor model. So red here are cases where the Gabor model predicts better than the deep neural network model, and blue is vice versa, and you see everything here is white and blue. So it's really a kind of a superior model. And you can compare them by plotting prediction accuracy of the Gabor model on the x-axis, the network model on the y-axis. This is the line at unity, and each one of these dots is a vertex in one of these maps. And you can see that there's just a real advantage to using deep neural networks to try to predict brain activity. And by the way, this works for mice too. That's interesting. Now, in terms of alignment of brain areas with the deep neural network layers, what we've done here is to color the map now, not according to prediction accuracy, but use a color code that indicates the layer of a deep neural network that is most predictive of brain activity at each point on the cortical sheet. Here are the early layers like V1, and you see that the early brain areas like V1 are best predicted by early layers in the visual cortex and neural networks, sorry. And as you progress towards deeper layers from the posterior, the anterior direction in visual cortex, the best predicting layers are deeper in the neural network. Okay. So this is really compelling evidence, I think, or can be interpreted as compelling evidence that V1 through at least V4 and beyond form a kind of processing hierarchy, because I have a model that really is unequivocally a processing hierarchy. It's a series of function compositions. It's doing a great job of predicting brain activity. It solved a hard visual problem that we know that brains can solve. And maybe most importantly, it orders brain areas according to the hierarchy that's established by the network. But I think it's important to be critical of this finding. These networks have thousands, maybe millions of parameters. And it's not at all clear when you get a result like this, why the model is achieving the success that it is achieved. So I want to think critically about why or if the alignment between deep neural networks and brains provides evidence that the primate visual system is a processing hierarchy. And that's going to start, I'm going to start that critique by pointing out But there's at least 3 kinds of hierarchy that are going on in the deep neural network. All right. So let's say we're reading out activity from 2 units in the brain, a gold unit and a blue unit. All right. And let's say that this is a deep neural network that can be used to accurately predict that brain activity or read out that brain activity. What I'm illustrating here with a compositional hierarchy, all it means is that you need more layers to predict the gold unit than you do the blue. So in some sense, the gold unit is more complicated or maybe more nonlinear, if you want to say that. And that is a notion of hierarchy that's kind of baked into deep neural network models. A distinct notion of hierarchy is what I call entailment. In this case, you have a deep neural network that's predicting the gold unit, and all of the layers and the network that you need to predict the gold unit can be used the bottom layers can be used to predict this blue unit. All right. One way of saying this is that if I have a model of the gold unit, I get a model of the blue unit for free. All right. But the other way around is not true. Right? All I need to predict the blue unit are these first 2 layers. I don't need the last. So that puts the gold unit higher on the entailment hierarchy than the blue. And finally, there's this notion of a spatial integration hierarchy. And that what I'm illustrating here is that the processing that leads to accurate readout of the gold unit pulls over 4 pixels, a wider window on the stimulus. It has a wider window on the stimulus than the blue unit, which puts the gold unit ahead of the blue in the spatial integration hierarchy. So all of these are at least possible or might be indicated when you have this result that aligns a deep neural network to brain activity, but they're all distinct. Okay? So compositional hierarchy that I've drawn here is not an entailment hierarchy. The entailment hierarchy is a compositional hierarchy. It's probably a spatial integration hierarchy, but the illustration I'm providing here is not an entailment hierarchy. So these are completely distinct ideas. They're kind of conflated in most deep neural network models. And it's not clear which, if any of these, is really important for getting accurate predictions of brain activity. It's not obvious. That's what I'm going to address now. I'm going to show you a series of analyses that tries to answer this question. Which kinds of hierarchy, if any, are actually needed to get accurate prediction of brain activity in V1 through V4? And here's the logic of our approach. Let's say we build 2 models. Okay. One looks like this. It's got a single deep neural network, single branch deep neural network, and we're going to read out the activity of our units in the brain from all of the layers. And if we had enough data, which we do, we could train this neural network and these readout connections to just be optimal predictors of brain activity. Right? And if any one of these 3 kinds of hierarchy that I just discussed are important to the brain, then this system with its many thousands of free parameters will learn it. Okay. Now, if I take another system that has a totally different architecture, in this case, we have 2 branches, and each one is dedicated to a single brain area or a single unit in this illustration. But this system can also learn all 3 kinds of hierarchy as well. If I optimize this system to predict the brain activity, it should also learn any kind of hierarchy that the brain needs in order that the brain actually expresses. Right. So what we're going to do now is actually build models like this using lots of data. And the logic here is that every time you build a model and it shows evidence for 1 or more kinds of hierarchy, that strengthens the idea that it's important for understanding the brain. If you encounter one counterexample where these kinds of hierarchy don't show up in your model, that means they're not important. That's the kind of logic. We're going to pursue a proof by counterexample. All right, in order to build, and these are, by the way, the kind of first picture I'll draw, brain optimized network models. They're neural networks that are just trained to predict brain activity. They don't are not explicitly trained to solve any task, any computer vision task, I should say. Here's a more in-depth picture of 2 CNN-based models, and we call these encoding models. In general, that's a general term for any mapping between stimulus and brain activity in the visual system. right? Every encoding model consists of a deep neural network that's basically used as a feature extractor and a readout head, that in our case can sample from any one of these layers and applies a spatial receptive field and a set of feature weights in order to predict brain activity. So the stimulus goes in to the feature extractor, it's read out by these readout heads, and you get a prediction of brain activity. On the right, I'm showing a task optimized case. So the feature extractor is trained on a series of labeled images. There's no brain data involved in the training of the feature extractor, but the readout head is trained using brain data. In the brain optimized case, we have a feature extractor that we call GNet, and it is trained along with the readout head to predict brain activity. So this neural network knows something about labeled images, and this only knows about brains. Okay. The way we train all of this is in order to train models like this, you need a lot of data. And we recently released with a number of collaborators a data set. And this was really pioneered by Kendra Kay, called the NSD, or the Natural Scenes Data Set. It's 8 subjects. It's high resolution, relatively high resolution scanning fMRI, whole brain fMRI of 8 subjects, each of whom had seen tens of thousands of images. So we scan them basically every week for close to a year. So we have enough data now. And by data, I mean brain activation in response to an image. Thousands of these training pairs, tens of thousands, actually hundreds of thousands of these training pairs to build brain optimized networks. So it wasn't clear to us that these brain optimized networks would work initially. So I'm going to convince you that they do work very well. I'm going to present a sort of head-to-head competition between deep neural networks that are based on a task optimized network, in this case, AlexNet, for those of you who know, and a predictive encoding model that's based on GNet, right? And the difference here is all in the gears, right? What I'm showing here are the different elements that are actually trained using brain data versus the ones that are fixed. And really all that's fixed here is the AlexNet in this case. And what happens is we take data that have not been seen during training at all. We run them through this processing pipeline, and we get predictions that we can then compare just using simple Pearson correlation to the brain data, to the brain activity that we record in our 8 subjects. Okay. The way that we often show comparisons between 2 models is in an accuracy advantage plot, and I'll just explain that. What we have on the x-axis is the difference between the prediction accuracy of the AlexNet model and the GNet model for all of the voxels in one subject. Each dot is a voxel. If the voxel shows up to the right of this vertical dashed line, it just means that the GNet model is out predicting the AlexNet model. If it shows up to the left, it means the other way around. And the vertical height is just the average prediction accuracy of the 2 models. What we see for subject 1 is a really clear advantage, fairly clear advantage for the GNet model over the AlexNet. So something like 80% of the voxels are better predicted by this brain optimized model than the AlexNet model. If we now take out of sample images that again never seen during the training set, and we run those through, and so these are images like gratings and different kinds of noise, white noise, ink noise. We run those through our models and we get predictions. We see an even greater advantage for the GNet model, for even more. And this is just really consistent for both natural scenes and synthetic stimuli and across all eight subjects. So I'm not trying to make the case like we win, we have the best predicting model, right? I'm just trying to make the case that you can take a different approach. If you train your neural network just to predict brain activity, and maybe not surprisingly, you do just a little bit better at predicting brain activity than if your neural network was trained to solve a computer vision, right? What this does mean is now we can interrogate both models and ask them which kinds of hierarchy are important. Which kinds of hierarchy do you invoke in order to predict brain activity as accurately as you do? Nice thing about the brain optimized model is that it gives us a certain amount of flexibility. Okay? When you're working in a task optimized domain, you have to choose architectures that solve your task. Here we can choose kind of any architecture we like that will predict brain activity. And the two kinds of architectures we tested are the single branch, which I just showed results for, but we also tested a multi-branch, right? So in the single branch, you have one deep neural network and a separate readout head for every brain area in every section. In the multi-branch case, you have a single deep neural network for every brain area. It's dedicated to each brain area. And then you have also a separate readout head for every brain area in the subject. Now, if you interpret these deep neural networks literally as like each layer is a certain brain area, if you give it a literal and like anatomical interpretation, this model just grossly violates anatomy, right? It implies that you could knock out V1, for example, and leave activity in V2 through V4 completely unaffected, right? So they have wildly different interpretations in terms of anatomy, but as it turns out, they have more or less identical prediction accuracy. Okay, so I'm showing you again the accuracy advantage plot. This time color indicates subject, panels indicate different brain areas, and there's really no advantage one way or the other. You might have noticed that the range of the x-axis has shrunk from something like -2 to 2 in the previous plot. Now we're down 0.05. So they're really kind of at parity. So now we're in a position, we have two different models. They make different assumptions, or they have different implications about anatomy. They have different architectures. They're trained on the same data, and they make identical predictions. They have more or less identical prediction accuracy, and it's state of the art. We have not been able to build any models that do better than these brain optimized models. So now we can ask, which kinds of hierarchies show up in these models? Initially, we look at integration hierarchy. Right? If there's an integration hierarchy, what we expect is that as you progress from V1 through V4, you should get an expansion in receptive field size. And that's what we're showing here for both the single branch and the multi-branch model. On the x-axis, you have eccentricity, which is just how far away from the center of the visual field a receptive field is. And then on the y-axis, you have size, which is how big a window that voxel has, or that receptive field has. Each dot is a voxel, and color corresponds to different brain areas. And what you see, and this is a very kind of classic result, is this fanning out of the lines that relate size and eccentricity. In all areas, the farther you get away from the fovea, the larger your receptive field becomes. But across areas, at any point on the fovea, at any point on the visual field, you have much larger receptive fields in V4, for example, than in V1. And it doesn't matter if you use a single branch or multi-branch. So this strengthens the evidence. It strengthens the idea that integration hierarchy and expansion of receptive field size is probably important, an important organizing principle. What about this other idea of compositional hierarchy? If compositional hierarchy is important for processing in V1 through V4, then the network layers that are most predictive of V4 should be deeper than layers that are most important for predicting V1 through V3. Right? They're, as I said, more nonlinear, more complex in the sense that you need more of these nonlinear function compositions to predict it. We don't see evidence for that. So here again, we have the single branch and multi-branch models. On the x-axis, we have our 4 visual areas. Each bar is a different subject, so we're looking at all 8 subjects. And on the y-axis, we're just plotting the percentage of variance explained by the bottom half of the network. And what you see on the left is a kind of classic result. Lots of other groups before us have shown similar things, but the dependence of the lower layers in V1 is very strong, and it decays as you progress along the presumed visual hierarchy. Right? In the multi-branch case, this ordering of brain areas relative to depth just disappears. We don't see it. In fact, weirdly, in V4, you can explain almost all of the variance in V4 with the bottom half of the network, arguing that these extra layers we had that we gave the model that it could have used to predict V4 if it wanted to, were more or less ignored. Right? So this kind of really weakens the idea that the DNN brain alignment result is evidence for a processing hierarchy, because we see that the alignment of brain areas with depth, layer depth in the network, it's a idiosyncrasy. It just depends on how you train your model. It's not like a fundamental result. At least it doesn't seem like that to us. So what about this last? And this is kind of the strongest idea of hierarchy, entailment hierarchy. How do we test that? Well, again, entailment hierarchy is the idea that the neural network you need to predict V4 should contain all of the processing that you need to predict V3, which should contain all of the processing you need to predict V2 and V1, but it doesn't go the other way around. So if that's the case, then if you take a V4 model and you use it to predict V1, and then you compare those prediction accuracy to a V1 model, and that is being used to predict V1, they should be roughly a parity. Right? You do the comparison the other way around, in other words, take a V1 model and predict V4, it should do much worse than a V4 model that's been optimized to predict V4. Okay, and I said a lot of words, and I'll talk you through the logic in pictures. So we start with a model that is multi-branch. So we have a separate neural network for each brain area, okay, and a separate readout head as well. We're going to train this up on our data. Now, we are going to fix the optimized branch, the branch that's optimized for, in this case, V4. We're going to fix it. And then we're going to retrain our readout heads to predict V1. So now you have a V4 model that you're now using to predict V1. Okay? So we call this the prediction accuracy when the training and the readout heads are matched. And this is the prediction accuracy when the training and readout heads are crossed. Okay? And what you can do now is create a matrix. On the column, we have model trained on, so that's this case. And on the across the rows, sorry, on the rows, we have model trained on. Across the column, we have model tested on. And we're going to plot the difference between the two prediction accuracies that you get. Right? When training, when the brain area that you're trained on and predicted on are matched versus when they're crossed. Now, in a strict hierarchy, strict entailment hierarchy, so if we plot places where error accumulates, okay, comparisons or cross generalizations where there's error. In other words, the cross prediction is lower than the match. You would expect to see all of the error accumulate below this diagonal line, and you wouldn't have any error above it. In a completely flat, and we can quantify this kind of diagonal or this asymmetry with a number alpha. I can talk about how to do that. Right? In a flat case, you would see error accumulate on both sides, some amount of error accumulate on both sides of the diagonal. Because learning about V4 doesn't tell you any more about V1 than learning about V1 tells you about V4. All right. So now we can apply this transfer learning test to our various models and see what happens. When you apply this model to the single branch GNet, you get really strong evidence for an entailment hierarchy. Okay. So these are 8 subjects here, and this is the average across all subjects. And blue is error accumulation. So the cross validation prediction accuracy is lower than the matched. Right? And you see that all this error is accumulating below the diagonal, and this, recall, was the matrix we expected to see in an ideal hierarchy. However, when you apply this analysis to the multi-branch model, which has the same prediction accuracy, just a different architecture, evidence for entailment hierarchy vanishes. The representations look completely flat. Okay. Again, this is the ideal matrix you would expect, it's the matrix you would expect in the case of ideal flags. All right, so cross prediction loss is symmetric, which is not consistent with entailment. So here's an interim summary of the empirical results. We asked the question, what kinds of hierarchy are needed to predict activity with state-of-the-art prediction accuracy? And we found that integration hierarchy was important in the sense that all of the models we tried gave us an expansion of receptive field sizes. Right? Like basically any model we tried with the that's good. We saw no evidence that compositional hierarchy is essential. It shows up in some models, it doesn't show up in others. We saw no evidence that entailment hierarchy is essential. It shows up in some models and not others. So what can we conclude about our original question? So we started by trying to answer the question, why do we have diverse representations? And we were led to this idea of hierarchy. What I tried to show is that hierarchy is really multifaceted. It has it's just a word, but it refers to lots of different results in the literature that are not, in my opinion, they've not been really coherently related to one another, all of them at least. And by doing these brain optimized studies, we show that some forms of hierarchy, some important forms of hierarchy are not needed in the deep neural network model in order to permit state-of-the-art accuracy in terms of reading out brain activity. So, the answer to why do we have diverse representations, the processing hierarchy answer was that, well, it reflects a constraint. It reflects a constraint on local processing. You need multiple brain areas because a single synapse or a single neurons in a single area can't do all the work. And we just don't see any evidence for that. So this relieves open the possibility that diverse representations in the primary visual system have diverse functions. They're not rungs on a ladder necessarily, but maybe the standalone tools. All right. Maybe a more succinct way of saying this or expressing what I've learned rather from this work. I am less convinced that diagrams like this, which you see everywhere, they're at least formally equivalent to this, say anything more true about the visual system than a diagram like that, in which you have 4 visual areas and they're encoding the stimulus is expressed by 4 distinct functions. They don't necessarily relate to one another in a hierarchical fashion. The other thing I've learned from this is that, I don't know if learned, but the other thing that this is kind of direction this has pushed us in is to kind of get away from looking to the architecture of deep neural networks for insight into the architecture of the brain. Our experience with these kinds of models is that the relationship between artificial neural network architecture, and just representations in the brain, much less anatomy, is really loose and flexible and tenuous. It's really hard to interpret or to infer strong links between these things. So then what do you use deep neural network models for? I mean, it's fine. They give you great predictions. And I've done some deconstructing here. I've shown that one idea that is kind of conventional is maybe not as powerful as we thought it was, but what can you do with these models? And one of the directions we've been looking is to use these models to probe neural manifolds. So I'm going to show you a figure from a paper that was influential for me. It kind of turned me on to this idea of a neural manifold. What is it? Okay, so consider a space where every axis is an individual neuron, or in our case, a voxel. Now, you're going to present many stimuli to the brain, and you're going to record activity. And every time you record activity from many different neurons or many different voxels, I'll just say units, you're going to get a point in this space, right? So here, I mean, they're trying to sort of demonstrate that you always have more than 3 neurons. You have hundreds or thousands, but you can't draw a 1,000-dimensional space. So they're just making the point with these extra arrows that there's many dimensions. But in this case, let's say you recorded activity from this neuron, neuron 1, neuron 2, neuron 3, and you got many points in this space. So this paper, the figure that this paper is from is called Untangling Invariant Object Recognition. And one of the points it makes is that there is, first of all, that if you show all of the stimuli from a single concept, let's say all the pictures of spaces that you might be able to show, that the points that in activation space that are evoked by all these stimuli, they're going to collectively sweep out a manifold. And a manifold you can just think of as a collection of points and activity space, and they're sort of illustrating them as 2D sheets in a 3D space. All right. But they're just trying to draw these kind of weirdly shaped structures. And every point on this manifold is the brain's response in V1 in this illustration to a single kind of stimulus. And every point on the red manifold is maybe a response to some other kind of stimulus. They made the point that it's not only that if you want to separate, let's say these were pictures of faces and these were pictures of dogs. If you want to be able to distinguish between them, these manifolds have to be separated in activity space. They can't be all bunched up. Right? Otherwise, if a point belongs on both manifolds, it's hard to tell the difference between them. He also made the point in this, I think, pretty cool paper that it's not just the amount of separation between concepts or object manifolds that's important, it's also the way that they extend in the space. Because there's going to be many thousands, tens of thousands of objects, or maybe an indefinite number of objects that you want to discriminate between in your lifetime. So these manifolds have to be very carefully arranged with respect to one another in order to permit accurate discrimination. And they pointed out that there had been at the time, and it still kind of exists, this long-standing idea that neurons deep in the visual system, like in IT, are kind of point manifolds in their parlance. Single dots in this space. And let's say if you present various pictures of faces, no matter how the exemplars of that face vary, you just get a single point. Okay, so you get a response whenever there's a face and no response when there isn't. Okay. This way of thinking about representation is very different from the one that I spent the first 50 minutes talking about. And it raises a new set of questions that are really interesting to explore. And that is what geometric properties change across visual areas. I've just shown an example that there are diverse representations across V1 to V4, but I haven't said anything about the geometry of the neural manifolds. And the other question is, how do these changes in geometric properties affect representations in each area? So we've been looking at this, and we've been guided by a theory called that was published in this paper that's currently on bioRxiv. I'm assuming it's in review. The paper is called The Geometry of Concept Learning, that relates manifold geometry to a very specific and important function that our brains solve. And that is classifying new concepts from very few examples. This is something that humans do really well, even very young humans. And I'll just illustrate it. So let's say you have, in your life, you've seen some small number of pictures of raccoons and some small number of pictures of wolverines. Let's say it's just 3. All right. Now I can tell you that in making this slide, I doubled the number of wolverine pictures that I have ever seen. Right. So this is a new idea to me, or a new object category of this. And in few concept learning, the idea is that you have a system where after only seeing 2 or 3 examples or some small number, let's say M examples of 2 different categories, you can be presented with a test example and get the answer. So I don't know if any of you have ever seen a wolverine, but if you haven't, it doesn't matter. You all know that those are raccoons, even though there are 5 raccoons in that picture and only 1 on the other. You've already developed, just by boom, looking at the screen, a robust representation of the difference between these 2 categories. And it accommodates lots of variation in the way that these object types present themselves. This is something that is much harder to train into a neural network than into brains. Okay? People seem to do it effortlessly. So how does this relate to the geometric properties of neural manifolds? Well, that's the work that this really interesting paper does. It identifies at least four geometric properties that are directly related to the generalization error in this few-shot learning context. One of them is, okay, so in this case, in this theory, instead of thinking of manifolds as sheets, these flat sheets, they model them as ellipsoids, high-dimensional ellipsoids. Okay, and that is it's an assumption that may or may not be valid, may or may not be true, but that's what they do, right? And they start off by showing what is the effect of different geometric properties of a pairing of two concept manifolds, A in blue and B in green. And they start off by saying, you know, we want to know how accurately we could discriminate, or a downstream neuron could discriminate between the A and the B concept, or let's say object category, if manifolds were structured like this. Okay, so here they're showing one of the important considerations, geometric considerations, is what they call signal, and that's just how far apart these manifolds are. And we already kind of saw an example of that in the previous video. And what they show is that as these manifolds come apart, right, their signal, according to them, increases. So signal is basically just a normalized measure of the distance between the centers of these manifolds. And generalization error decreases really rapidly, even if you have n, which is the number of samples, just one sample. They identify another potentially important property, and that's the relative sizes. Okay? When one size is larger than the other, you get varying effects on generalization error, but in general, as you vary bias from bias, it's basically a matter of bias as a measure of the relative radius of the two manifolds. And as you increase one relative to the size of the other, you advantage generalization error for the different concepts. Another important property is dimension. I don't want to go, they have a very kind of detailed definition of manifold dimension, that as it increases, generalization error goes down. And then they have another idea, which is single noise overlap, which is the idea that if the vector that separates the centers of these two manifolds overlap at their extents, their orientations, you're going to have a problem discriminating them. So as this overlap increases, generalization error goes up. So each one of these aspects of manifolds are kind of abstract. You can't actually look at these things. You can only calculate these statistics. One really nice thing about their theory is that they put all of these things together. And they show that if there's a specific expression that combines, it looks like a signal to noise ratio effectively, that combines signal, bias, dimension, and overlap. And it expresses, it's expression that is an explicit function of the number of samples. And it actually just very accurately predicts generalization error. So you can, by measuring these properties of neural manifolds, you can know if the theory is correct, without even doing this classification test, how well a brain that has these manifolds in them would do with the few-shot classification problem. Right? So we decided to use our brain-optimized models to probe this aspect of neural geometry, okay, neural manifold geometry. And we basically did another simulated experiment. Okay, so Gislan took the images from another database called the ImageNet and just ran thousands of examples of different concepts through the model, which gave us an ability to probe noiseless representations of neural manifolds. Okay, so we have a manifold for cats and a manifold for dogs and a manifold for cars, etc. And we can run as many samples as we want through this, and that will give us, and then we can calculate these statistics that I just outlined. First of all, so we want to show whether, I mean, this simulated experiment allowed us to test how well our brain optimized networks solve this few-shot classification problem. And here we're showing 5-shot classification error. So you're building a linear classifier based on 5 samples from 2 different categories, right? And you're doing this thousands of times for thousands of pairs of categories. We're showing how error scales as a function of semantic distance. Okay, so and we measured semantic distance using WordNet, which I can go into, but it basically, and semantic distance is like two different kinds of dogs are going to be close, but dogs and cars are going to be very far away on this x-axis. And for sufficiently semantically distinct objects, you can get really small amounts of 5-shot classification error out of your models of higher level visual units. That's interesting. It kind of reverses the analysis direction that's typical. Instead of starting with a deep neural network that solves a computer vision problem using it to predict the brain, start with a deep neural network that predicts the brain and used it to solve a computer vision problem, and it does reasonably well. Interestingly, the theory, the geometric theory that they propose in this Sorcerer et al. Paper, fits and predicts the classification error really well. Okay? So we can go in and measure these statistical properties, these geometric properties of the neural manifolds for every concept. And we're able to use the theory to recover the actual empirical classification error for all of these brain areas. Now we're in a position to track how the geometry of these neural manifolds changes across brain areas. And one kind of surprising thing. So in each one of these panels, we have the different brain areas aligned on the x-axis, and we have changes in SNR, which is the kind of total expression, which you can break down into a function of signal, bias, various kinds of overlap and dimensionality. And the only thing driving an increase in SNR, which ultimately determines your few-shot classification, is an increase in signal across brain areas. All of these other measures either are flat or they change in a way that actually would drive classification error down. So the geometry of these manifolds is not changing in a coordinated way across brain areas. Some aspects of them are advantaging this particular task, but some aspects of them are mitigating that advantage. It's possible that they're changing in a way that optimizes some other tasks. We don't know. The other kind of interesting thing is that dimensionality does not change monotonically. And we've seen this now and looking at the data in a number of different ways. There's actually an expansion of dimension from V1 to V2, and depending on how you look at it, that expansion sometimes lasts a little bit longer into the hierarchy. And then this really graceful decrease in dimension. Again, so this again is kind of leads us to some kind of interesting conclusions. And I'll try to summarize both parts of the talk. So many visual areas may exist, serve many different purposes. That's my hedging answer to the first question that I posed. If you want to think about the primate visual system as a hierarchical progression, I think it's important to know it's a very limiting view of primate visual processing. I think it underestimates probably the talent of individual neurons, and it was very weakly supported. It's not supported at all by the results of our study. I'm sure there's other data out there, but in our study, it's not supported. Methodologically, I've shown that with enough data, you can build brain-optimized networks. They achieve state-of-the-art prediction accuracy. They don't make any assumptions about visual function, but they do learn to solve visual tasks more or less for free. The relationship between architecture in these brain-optimized networks and brain representation is really loose and flexible. And I've come to the conclusion, or I've been led to think from these studies, that the success of any one deep neural network model in predicting brain activity, you can't conclude much from it. You have to be very careful in the inferences you draw. That said, I think that geometric properties, using these networks to probe geometric properties of neural manifolds, might be really revealing. That's a kind of a new direction, at least for us, and I think for the field. And I think brain-optimized encoding models are a really good tool for doing this, because they denoise activity and they make no assumptions really about what the function of these brain areas are. All right, so that is, I'm only 2 minutes. I'll finish with my acknowledgements and you can do questions.
Yeah.
Well, thanks a lot. Very impressive talk. Thank you for making me doubt the hierarchical model. It's been a long time since I've thought about this assumption we all have. I think When I was listening to you, I've been thinking about higher level vision for some time, and it got me to think, like, what do you see of these early visual region and their report to the rest of the brain? We always say that, well, top-down attention will boost signal in like these regions, or there's this paper by Di Carlo's lab that showed that essentially you need VMPFC's input to bring stuff to consciousness in monkey. So my question is, how do you see these other regions interacting with different layers of networks? Do you see that they interact at like some stages in particular, or not at all?
Yeah, my intuition from all this is that each one of these brain areas is optimized for a certain kind of, to solve a certain kind of task. And the fact that all of them are amenable to top-down, and we know of, they're all drivable and modifiable by top-down input, right? That to me suggests that other brain systems, the way I think about it at least, are reaching out to these areas and trying to extract information that each one of these areas maybe perhaps uniquely represents, right? So when I see tuning shifts due to attention or, I don't know, memory or expectation or mental imagery in a lower area than V1, it implies to me that those V1 representations are important to solve the task that these much deeper brain areas are changing or, interfacing with V1 to solve. So again, that kind of gets away from the idea of a hierarchy. I'm not saying that there isn't a hierarchy. I'm just saying that it's kind of unrelated to the idea of hierarchy. It's the idea that each one of these brain areas has kind of its own job to do. Another way of thinking about them is, I think, comes from this, suggested to me at least by this untangling paper, and that is that for certain tasks there may be an optimal point in the trade-off between the extension of a neural manifold and its separation from others. Okay? So if you really just wanted to separate concepts, you would have point manifolds that are invariant, okay, that are not sensitive to exemplar variations, like the different ways that a face can present itself to you. But there would be some brain area that just isn't sensitive to that, right? And that would give you really nice separation and great classification accuracy, right? But for some tasks, you actually want to represent exemplar variation, for example, the pose or the position of a face, right? That kind of information is not just conceptual high-level information that's actionable. I mean, you need to know how to grab things, right? You need to know how to move around in space. It is possible that these intermediate areas they may be perfectly capable, or the neurons in them may be perfectly capable of decoding objects directly from the retina. I don't know. I'm not asserting that. I'm just saying we don't know. We have no evidence that they can't do it, or that the neurons themselves are incapable of doing that. In fact, the most recent evidence I've seen that if you want to get a good model of input-output relationships, even in early areas of the dendrites to spiking in the neurons, you need a 7-layer neural network, right? So, it's possible that these areas are, not to anthropomorphize, but they're choosing to accept less manifold separation in order for more extensive manifolds. Anyway, those are the two kind of high-level ideas I have for what is going on in the early visual areas. If you're throwing out the idea that they're slowly building up toward object recognition, you have to have some other idea in place. That's all I have right
now. But you could still have weighted readout by some other area. That could achieve that, like, let's say V1 does a function, V2 receives from V1 does a function, V3, V4, and then you have PFC or whatever prefrontal region that reads out a distributed pattern in
Sure.
the visual brain.
Yeah.
But like weighting, the importance of each of these separate functions to successfully recognize something.
Yeah, yeah, I mean, again, though, I feel like that somewhat undermines the idea of the kind of pure processing hierarchy idea. It suggests that there's something about these brain areas that makes them amenable to recombination in order potentially to solve novel tasks. I'll take a little bit of what V1 is doing and a little bit of what V4 and some of IT.
So I suppose this leads me to the other question I had, which is, at some point you had your feed-forward model and you turned it around and then said maybe V1, V2, V3, V4 are simply doing different functions. And what I found curious was that there was no connections between V1, V2, V3, and V4 in that cartoon, right? So, I mean, it's not because they're doing separate functions that they wouldn't be able to learn from one another. And then the question really is, do you think that if there was some recurrence in your brain optimized models that allowed for the different layers to talk to one another, that you could get even better prediction?
Maybe, I don't know.
suppose it's
We haven't, yeah, so we've tried a number of different architectures. Different architectures don't make a huge difference. That I know of, at least, Siobhan has not tried optimizing recurrent architectures. I will say, though, if you look at like a recent paper by Kopitar and Jim DiCarlo's lab, when he was in Jim DiCarlo's lab, they went to great pains to isolate portions of IT response that were almost certainly due to feedback or recurrent processing. And they show that a really deep neural network can predict those responses just as well as a current one. And their logic, which I buy, is that, in one case, you're trading off cycles, substituting layers, vice versa. So this leads me to 2 points. One, there is a kind of, I don't think any of this really depends on whether or not there are recurrence or not in our models. A recurrent neural network can have compositional entailment and spatial integration hierarchies, and non-recurrent neural network can as well. And again, we're just looking for one counterexample that has state-of-the-art prediction accuracy and none of these kinds of hierarchy. And we found one relatively quickly. And then, I mean, the other thing about that is it really underscores, at least for me, that well, it emphasizes my current discomfort in really imputing any literal and like an anatomical interpretation to connections in a deep neural network. I mean, we know that all these brain areas are recurrently connected. In fact, the most well-established concept of hierarchy is the Feldman-Van Essen hierarchy. And it references layers and reciprocity. Those are the main defining concepts, and they were completely absent from deep neural networks, or at least the models that I've been talking, especially layers. Like there's no AI model with cortical layers that I'm aware of right now that imputes functions to them. So I'm really taking a step away from taking deep neural networks seriously as models that have a literal or mechanistic interpretation. To me, it seems safer right now to just think of them what they've always been, which is function approximators, really good ones. You give them enough data, they'll do anything you want. I mean, consider this too. If you have enough data, you could fit an individual neural network for every neuron or every voxel, right? And what would the anatomical interpretation of that model be? That all neurons have no connections to one another. But we know if you have enough data that the bespoke model for every neuron would predict as accurately as anything you could build, right? And the literal anatomical interpretation of that best predicting model would be nonsense. It just would have no basis in reality. And I think I'm just giving a case study in that thought experiment that we could have done all this work. I mean, I guess that's architecture.
Yeah, you said something quickly about the trade-off between the cycles and number of layers. And just to double click on that a little bit more, were you saying something to the effect that either side of that is equivalent, or that it would be preferable because it's more amenable to the current models to emphasize the number of layers and to play around with that idea rather than the cyclicity of it?
No, I was just pointing out that the former, that there's at least the logic that I understood in that paper, that there's a kind of equivalence between cycles and layers. In other words, I can get more applications of this nonlinear function by allowing my recurrent neural network to process for more cycles. In the same way that I can get more applications of the nonlinear function by putting more layers into the.
So there's no implicit favoring of either one model or another, I guess. Because
I'm not favoring one model over the other. Now, I will say though, I'm not looking at dynamics.
Yeah.
Right. So I mean, I hope that was clear. Like, if you want to account for dynamics, I don't see how you get away from recurrence. I mean, but I'm studying the response to static images, and maybe all of this goes away once you start really considering dynamics and moving images and things like that. I don't know how well any of these lessons that at least that we're drawing will generalize to that. Yes.
You were, for example, looked at the geometric characteristics that you sorted in task-oriented networks.
Yeah.
And some of them are actually predictive of few shots capacity.
Yeah.
AI models.
Yeah. Am I aware of that?
Yeah.
Yeah. So in this paper, actually, in this paper, they don't have fMRI data. They have some electrophysiological data that they study, but a lot of the results they are applying to layers of a deep neural network that was trained on ImageNet to do object classification. And somewhat surprisingly, they find that at least deeper in that neural network, the representations there support few shot learning really well, about as well, or maybe even better than the brain optimized networks we're showing here. Yeah, I mean, I said it's a harder, in general, few shot learning is a harder AA problem to solve than learning with millions of examples. But in fact, if you do have millions of examples, and once training is done with millions of examples, few shot becomes, in this case, and in their paper, it's pretty good.
I was also curious if your brain network is actually very good at short-term, but from what you said, I understand still not as good at a big artificial net that's task-oriented and trained on millions of them.
It's a good question, and I need to go and look. I need to go and look at the numbers they have in their paper, and actually the numbers that we have in models that we build in lab. I will just say that my memory is that these deep neural networks are achieving few shot classification, or at least in the deeper layers, that's below 5%. I don't recall the dependence on semantic distance. In our case, we're getting below, something that close to or below 5%, only for cases for pairs of objects that are pretty far apart on the word tree, right? Yeah, so that's why, I'm not claiming some kind of victory. I'm just pointing out that it's interesting that these brain optimized networks, they have learned something about solving visual tasks, which you would hope they would. Yeah, whether or not they're on par with systems that are trained with millions of examples to just solve visual tasks, I doubt it. But we haven't done like a kind of head-to-head comparison.
What's important is not the amount, but the reason behind the very different.
Yeah, it's a good point. That few shot classification accuracy and object recognition on ImageNet they're distinctly different problems. I mean, they're related in some sense, but it's just the anecdote that I have in mind to help me understand the difference is, I mean, let's say you're a museum curator and you have jars with thousands of different spiders, right? And it's your job to be able to look at any one spider and tell its species from the other, right? That is kind of like the ImageNet task, I think. But now let's say you're a tourist, you're going to, a forest that you've never been to for the first time. And your friend who's guiding you says, avoid that spider. And you only see it once. He's like, if you see that spider, never go near it. Right? That's few shot learning. And we have to pick up examples like that over and over. Every time we're in a new environment, we're stepping out of our museum. There are new kinds of objects that have new meanings and present themselves in different ways, and we have to learn them rapidly. And kids do that at a really young age. So it's an importantly different problem.